is Jacob, and I'm a PhD student in the Physiological Pharmaceutics group at the University of Copenhagen. In this video, I'm going to talk you through the use of in vitro transport studies across cell line monolayers, which is a very useful technique for the prediction of drug absorption.
There is a whole range of different cell lines available, each resembling specific physiological barriers. In the Physiological Pharmaceutics research group at the University of Copenhagen, we specialize in improving oral bioavailability either by absorption through the buccal or the intestinal barrier. The oral cavity can be mimicked with the use of TR146 cell lines, while the CACO2 cell line, which is the one we'll be using in this video, have many characteristics similar to the enterocytes lining the intestinal barrier. The experiment demonstrated in this video can thereby give an indication of whether a specific compound is either not able to get absorbed upon oral administration or whether it is. About 3 weeks prior to the transport study, CACO2 cells are seated on supporting filters, allowing them to differentiate. The monolayer then forms a barrier between two compartments, the apical side resembling the lumen of the small intestine and the basolateral side resembling the blood circulation. The transport study is initiated when the drug compound of interest is added to the apical side, and at predefined time points, samples are then collected from the basolateral side for determining whether absorption is taking place, and if so, at what rate. Depending on both the properties of the compound and the sample concentrations, different quantification methods can be applied. The flux is then determined as the slope of the linear part of the graph when plotting the cumulated transport over time. For more information regarding the concepts of flux and calculation of permeability, please follow the link in the video description below. Even though these in vitro transport studies can give a good indication of whether in vivo absorption is likely to occur or not, there are both pros and cons regarding their use. The advantages and disadvantages are both linked to the fact that in vitro transport studies are extremely simplified versions of the physiological conditions. This allows for a high throughput and relatively low expense compared to using animals. On the other hand, there is not always a strong correlation between the results obtained from cell monolayers and animal studies. It is therefore important to remember that these studies cannot stand alone, but is a useful tool in predicting and ranking different compounds based on their permeability. In the Physiological Pharmaceutics group at the University of Copenhagen, we're interested in improving the absorption of low solubility and/or low permeability compounds. By improving the solubility of a drug, for example by lipid-based formulations, more of the drug will be accessible for transport through the barrier, which would result in higher flux. Likewise, by increasing drug permeability, for example by formulation with permeation enhancers, increased transport of, for example, peptides can be achieved. As a result, steeper transport profiles will be observed when carrying out this study. Determining the integrity of the monolayer is an important aspect to include when carrying out transport studies. This can be done with a portable voltmeter with two electrodes, one immersed in the apical side and one in the basolateral, thereby measuring the transepithelial electrical resistance, the so-called tear value. The two most common instruments are the chopstick electrodes seen on the left and the endome chamber seen on the right. The latter will be the one used in the following experiment. As many permeation enhancers act by temporary reduction of the monolayer integrity, a decrease in the tear value can be a desired effect. On the other hand, if the mechanism of interest is through transcellular transport, a stable tear value throughout the experiment is preferred. For the following demonstration, the depicted equipment will be used. Most importantly, the CACO2 cells seated about 21 days earlier, portable voltmeter for tear measurements, transport buffer both for washing the cells before and after the experiment, transport buffer with the compound or formulation of interest for addition to the apical side, Pasteur pipette attached to a pump for easy removal of media when washing the cells, pipettes and pipette tips both for the addition of exact basolateral and apical volumes as well as for sample removal, labeled sample tubes for each well and time point, and lastly tweezers for handling the transwell when measuring tear values. With that sorted out, we're now ready to conduct the experiment.